Welcome, everybody. We're going to wait a few seconds to allow everybody to join the webinar. Okay, let's get started. So welcome to a webinar from Design Therapeutics. We are delighted to provide this information to you, and thank you for joining us tonight. This webinar is being recorded. If you have to log off for any reason, we will be posting the recording later through the FARA website and through social media, so you will be able to still get the information. I'd like to introduce you to our colleagues at Design Therapeutics. Depending on how you're seeing your screen, towards the top, we have Matt Kurth. He is the VP of Clinical Development at Design Therapeutics. We have Giles Seifert, who is the CEO of Design Therapeutics. And also joining us is Wen Nguyen, Senior Director of Clinical Operations. Tonight, during the webinar, you will hear a little bit about the mechanism of action of GeneTax, which is the therapeutic approach being developed by Design Therapeutics. We'll also be discussing the progress that is being made in the development of this treatment approach, and we'll end the webinar with a description of the proposed design for the upcoming planned clinical trial. We'll be taking questions at the end of the webinar. At the bottom of your screen, if you hover your arrow over, you should see a feature for Q&A. Please go ahead and enter any questions you have in that Q&A box, and we'll be monitoring it and presenting those questions to the design team at the end of the webinar. Thank you. And Matt, I'm going to turn it over to you to educate us.
Susan, thank you very much for giving us the opportunity to describe our progress in developing novel class of genomic medicines for genetic disorders. Also want to thank Jen Farmer for making this opportunity available. And let me put a pointer in place so that I can point out things as we move along. Hold on. As you may know, Design Therapeutics is a public company, and therefore when we come out with forward-looking statements, we must have a disclaimer. For those so inclined, this is also available on our website. I will not read it in detail. Design Therapeutics is based in San Diego, California, actually in Carlsbad, which is slightly north of San Diego, and was founded in 2017. We have about 75 employees, and we're developing a novel class of small molecules called GeneTac chimeras. And these are unique molecules that are truly chemically made, and they're not genes or gene therapies. They're unique small molecules. These molecules focus on serious genetic disorders by targeting diseases caused by DNA nucleotide repeat expansions. Our leading program is in Friedreich's ataxia, followed by the myotonic dystrophy type 1. Now, to give you an idea of what these molecules can do, let's review briefly a little bit about Friedreich's ataxia. As you know, Friedreich's ataxia is a multi-systems disease caused by the reduction of frataxin protein, which normally resides in the mitochondria and is an important part of the energy generating capacity of the mitochondria that's found in every one of our cells. The actual reason for having reduced frataxin expression is that patients have two expanded copies of an intronic GAA triplicate repeat that literally acts as a brake to the ability to convert the DNA information into RNA information. The result is that there is significantly reduced amount of frataxin in patients with Friedreich's ataxia disorder, as seen on the right side on top. Whereas compared to the normal expression of the gene, you see full expression of the frataxin RNA, and that converts, of course, into the frataxin protein. Our small molecules at very low concentrations can normalize the levels of frataxin RNA in cells from patients with Friedreich's ataxia or immortalized cells from patients with Friedreich's ataxia. Of note, these molecules have no effect on the frataxin levels in unaltered healthy cells. So what that means is that the molecules can literally unblock the block that occurs in the region of the frataxin genome where the repeats act as a brake to the ability for the polymerase to progress through that region. Now, how do these molecules do that? The mode of action is quite interesting in that it is a heterobifunctional molecule where one end of the molecule binds to the DNA and very specifically into the region that has the GAA triplicate repeat. There's a linker, and then there's a ligand moiety, another part of the molecule that's designed to literally help the RNA polymerase walk through that region of triplicate repeats and allow a full-length transcript to be made of the RNA precursor called, or the messenger RNA precursor, which is then spliced to make a normal RNA that's able to do all the functional activity required within the cells of the patient. What's important is that the frataxin that is made is completely the patient's own. It is not an exogenous frataxin. Now, what's also very exciting is that we don't have to be 100% able to return full frataxin levels. As long as we approach the levels of frataxin seen in patients who carry the mutations, the cells appear to return to normal function because we know patients who carry the abnormal gene are completely asymptomatic if they only have one gene. Only if they have homozygous frataxin mutations do you have symptoms. And the hope is that these molecules will allow the frataxin levels to reach at least into, if not to normalize, the cell frataxin levels that will hopefully restore functionality in this individual cells. Now let's talk a little bit about how far we've come along in developing these molecules. One of the most important things that is really that I'd like to impart to mention is the fact that these molecules only work in cells that have the actual frataxin gene abnormality. And thus, we've been very dependent on donations of blood cells from patients throughout the United States, and we're very thankful for that. We've also had patients that have been immortalized and then can be converted into cardiomyocytes and neurons. And we can find out that these gene-tagged molecules actually help return frataxin levels in those cells as well. We've gone through a number of iterations of manufacturing proteins and of making these chemicals, and we found one chemical that we think is a good candidate for bringing to the clinic. That candidate has been taken into animal models or animals, because there's no animal model of frataxin deficiency. And in those animals, we've been able to measure the distribution and pharmacokinetics of giving the molecule intravenously. The molecule does require intravenous injection, but it's not an infusion. The injections usually take a few minutes to occur, and then the molecules are rapidly distributed throughout the body, but last a long time as well. And I'll get into more of that in a moment. We've completed some non-GLP toxicology studies in 2 species, that is mice and rats, to show that the molecules and the molecule is well tolerated with multiple doses. In progress are FDA-specified GLP toxicology and pharmacological studies, which we've now are taking place and have been completed to a large degree in both the rat and non-human primates. Non-human primates simply means a small monkey that has very similar physiological characteristics to humans and is accepted by the FDA as a species that will allow us to demonstrate that the molecule is safe enough to start to administer into patients at the appropriate low doses, starting doses. Our hope is to have our first trial up and running by the middle or before the middle of next year. Now let's go over a little bit of some of the exciting features of the molecules. Here we have on the left the levels of messenger RNA after between 3 and 14 days of very low dose treatment. This is a drug given at 10 nanomoles. That's an incredibly small amount. And as you can see, the messenger RNA levels are increasing nicely. And these are tenfold change from baseline in relationship to DMSO placebo control. A few days later, on the right side, you can see that at the same level of 10 nanomolar with GeneTAC treatment, we begin to see the production of frataxin protein approximating almost the level seen in wild-type neurons, in other words, almost normalized. What's even more exciting is that the drug appears to go into all the important target tissues. So we find that we have data on the right side for brainstem, cerebrum, and cerebellum in blue, orange, and green. And after 3 treatments, 1 week apart, their level of drug remains above the 10 nanomolar level, which we showed earlier, to induce almost normalization of frataxin levels for up to 168 hours after stopping the treatment with the drug. And that's for both all the brain regions as well as the heart, as illustrated in the bottom right section of the panel. Thus, this molecule has the potential to reach all the target tissues that we want to reach, and hopefully can there, in those tissues, normalize the frataxin level in patients with Friedreich's ataxia. Of course, that needs to be determined. Now, last but not least, the drug has to be well tolerated. And when we look at the safety profile, so far in all the GLP studies in rodents and non-human primates, we've shown that multi-dose systemic administration of the GeneTAC molecule candidate has been well tolerated after 3 doses, 1 week apart. And that means that we are able to have the data that will allow us to submit an application to the FDA called an IND to start phase 1 trials, hopefully in the first half of next year. Now, before we get into the humans, we have to do a number of other things. And I just wanted to go back a little bit and describe the approach to getting these drugs into humans, into what we call phase 1 or first in human trials. And obviously, we have to have sufficient drug, but more importantly, the interaction between the body and the drug has to be somewhat understood. And we use animal data from the mice, rats, and monkeys to figure out how well the drug is absorbed. In this case, we're using a small injection into veins, how it's distributed throughout the body, how it's metabolized, and how it's excreted. Given that information in animals, we can then actually model how it might behave in humans to allow us to predict the starting dose as per FDA guidance and how long it might stay in the body, so that we can estimate when to take the appropriate blood samples and do the studies in the human population, hopefully that will allow us to move forward from phase 1 to phase 2, etc. As I mentioned earlier, the drugs, the GeneTAC molecules, only work in patients that actually have the GAA triplicate repeats. And so we must turn to humans and straight into patients to understand how the drug interacts with the pharmacological target, how the molecular response occurs, in other words, how well and fast the frataxin protein levels increase, and then to see directly or indirectly how the cells and organ tissue subsequently respond. Now, a few words on some of the general inclusion and exclusion criteria. And to make a long story short, these are very, very non-specific at this point. There may be some additional details, but because we are looking at safety first, the majority of the inclusion criteria really concerns having patients who are healthy enough that they can give adequate information on the safety of the drug. And so these would be male and females, 18 to 55 years old. FDA has very strict requirements as to how to enroll younger individuals than the age of 18, and that will require additional work with animal toxicology studies, as well as having some preliminary safety data in adults. Obviously, the drug only works when you have GAA repeat expansions present, so that's a criteria that we need. And because the drug will be looked at from a pharmacokinetic perspective, in other words, how fast it goes into the body, how long it lasts, and what kind of an effect it has on peripheral blood frataxin levels. Generally, if patients can if patients can transfer themselves is probably and safely participate in the study, then they should be eligible for participation. Again, just to point out, the primary outcome will be safety as measured by adverse events, laboratory tests, and cardiac monitoring. There will be significant amount of looking at the heart function to make sure that patients can tolerate the injection of the medication. And then secondary, we'll look at the levels of the drug in the bloodstream, as well as the frataxin levels in white blood cells. The FDA has very strong guidance. They like to see double-blind placebo-controlled trials. They like to have what's called a sentinel dosing, which means that before a group of patients fully are treated, we start with one active and one placebo patient, see how they are tolerating the drug over the first few hours and maybe a day or so. And that data is always reviewed and before enrolling the rest of the individuals. At the end of each cohort, and in this case, the proposal would be 5, but it could be more, could be less. The dosing will be evaluated by a safety review committee that looks carefully at all the data that's being collected to make sure that the drug is well tolerated. The starting dose will be determined by adverse events found in animals, and we have to go one low dose below that. And there are some other rules and regulations that will be followed in terms of the mathematical formula to determine the exact starting dose. It will be, it's based on what's considered to be a safe starting dose, and it may not be at the level at which the drug is actually efficacious at this point. It may actually begin below the level of efficacy. The second cohort and the intervals between cohorts can be done using a variety of different rules, none of which, and that has not yet been fully determined. But the plan is to have 5 cohorts where the middle cohort should definitely be within what will hopefully elicit an appropriate response at the blood cells that we will use to measure frataxin, messenger RNA, and protein levels. Once the phase 1 single dose study is completed and the data is considered adequate to be able to move forward to a multi-dose, and we have sufficient data in animals to show that it's safe to give in multiple doses, then another study will be started, which will allow at least 3 doses to be given. And again, 5 cohorts with 5 patients per cohort will be probably proposed. The inclusion criteria and exclusion criteria should be very similar, and similar pharmacokinetic and pharmacodynamic assessments will be considered and performed. Initially, the proposal is for 3 weekly doses, so that the study will take approximately 3 to 5 weeks to complete. And it is this dosing that will allow us to determine what might be an appropriate set of doses to be used moving forward into the next set of studies where we will look for real clinical benefit in the phase 2 program. And hopefully that gives you an overview of where we are, how the drug works, and what our plans are to get into the clinic and move the drug into a true evaluation for its benefit in patients with Friedreich's ataxia. Thank you very much for your interest in listening, and I think I'll open it for questions right now. Thank you. I'll stop sharing unless we have to go back to a slide.
Well, yeah, I would keep the slides up. I think there's some questions that will help with clarification. Thank you very, very much for your presentation. One of the first questions that came in that you may have mentioned, and we could just use some clarification. The question was around maximum number of GA repeats that might limit the function of GeneTACs. Is there an upper limit for that repeat expansion?
Joel, I'm going to turn it over to you because that's a detail. I'm not aware of any, but I'm going to defer to you on that one.
Yeah, correct. Obviously, we haven't tested all the entire universe, but within the general distribution of the FA population, ranging from, say, low 100 repeats to over 1,500 repeats, and we tested a very large number of patients so far. and the response has been quite comparable across the repeat length. So the answer is no. Simple answer is no. Not that we could detect.
Okay, thank you.
There's another question that I think builds on that, right? There was a question also about the gene tag mechanism, and is it specific to the GAA repeat, and could it have any off-target effects on other repeats in the genome?
Yeah, that's an important question, one that, of course, we looked at very carefully from the outset, and it's much of the scientific work addressing this question is in the publication from Dr. Ansari, our co-founder, back in 2017. So there are various levels of the way the gene tags work that confer very good specificity to the GA repeat. First, because GA repeats elsewhere in the genome usually are not associated with initiation of transcription, certainly not for other genes. And if there is no mutation, the transcription is not blocked. So the only place in the genome that has a block in transcription and where transcription initiation is happening from a GA repeat is in the frataxin gene. The gene tags do not alter initiation of transcription. They simply unblock a transcription elongation that is blocked in the case of the long GA repeat expansion. So in that sense, when they looked at the broad RNA-seq, and getting to sort of technical speak here, and this is published data, they did not see perturbation of the broad genome. And this is quite consistent with the way the molecules work.
We've had a couple of questions come in, which we expected, asking about any studies that may have been done or may be planned around pediatric or offering a pediatric arm to the studies that you are planning.
Yeah,
Yes.
The FDA has specific guidance as to how to start pediatric studies. Number one, they would like to see some data in adults to show that it has safety. And then there's a series of preclinical toxicology studies that have to be run in animals that are immature to show that it's safe in that setting as well. And that's because the metabolism may be different in immature animals, and that would, of course, might be true for humans as well. But we're definitely needing to collect the data for adult safety first, and then we can, as well as move forward with the preclinical toxicology package.
Okay, thank you. And so along with that, not pediatric, but in terms of participants who would be eligible to participate in the phase 1 SAD or MAD, will there be an open label extension planned?
Not at this time, given that for now the exposure is relatively short and the progress. We're trying to move as fast as we can to answer two main questions. One, of course, whether this molecule is safe in people, and that's critical and foremost. And second, whether we can detect an increase in frataxin in people who receive gene tags. In parallel, and of course, these studies continue to be in motion internally, we are extending the toxicology assessment of these molecules over a longer period of time that then, in effect, could be used for running longer studies. But I think it's very important to get a first read of the tolerability and ability to restore frataxin in humans as we are beginning our development.
Great, thank you.
And I think a follow-on to that is a question I see that asks, how are you going to measure frataxin during the clinical trial? Is that in peripheral tissues like blood and skin?
Yeah, so we intend to measure frataxin in peripheral blood cells. These are fairly well documented and very well defined. Of course, to do this in the clinic, we have to do this at a clinical level of scrutiny, and this is all underway.
Yeah.
And because the gene tags are otherwise unrelated to anything to do with frataxin, right? They're just standard small molecules that elicit endogenous frataxin. And this, I think, is an important, perhaps, point that may be not so clear from when we speak about this. The gene tags elicit a restoration of the endogenous frataxin production, and that's important. By endogenous, I mean the body's own machinery, the cell's own machinery, are then enabled to make more frataxin. Of course, even in individuals with Friedreich ataxia, there's still a certain amount of frataxin that is made, it's just not sufficient amounts. And what gene tags allow is to unblock the transcription so more frataxin can be made. And this frataxin that is made by the body's own machinery, and it's regulated by the cell's own machinery. So it's not only produced by the own cells, but also regulated by the individual's own system. And I think that's an important feature of this approach.
Yeah, and I think a follow-up to that is another question here where somebody's asking does the frataxin stay in the body or in the cell, the frataxin that gets made, or does it go away again?
Yeah, so frataxin is probably some of the community know well. Frataxin is not a protein that is secreted. Once it's produced in the cytoplasm, it travels into the mitochondria where it's supposed to reside. And we know from published literature that the frataxin sticks around for several days sometimes. So the gene tag, so think of this way, gene tag will unblock transcription and produces these RNAs, keeps producing them as so long as the gene tags are present. And we noticed, and Matt showed us, that the gene tags remain present at a very good concentration for over a week after a dose. So of course, then a week later, you can redose gene tags again and so forth. While the gene tag is present, you have transcription of the DNA making the RNA that gives the instructions for production of frataxin protein. So it's sort of a cascade that keeps sort of a self, renewing, because a molecule of RNA can make many, many molecules of protein. And the protein itself, in turn, lasts for several days. So this enables what we'll call a pharmacodynamic effect, or an effect, from the initial intervention on to make protein that remains in the body for quite a while.
Thank you.
There's a couple of people that are asking questions about the study design. Are you able to discuss or disclose whether the study will be US-based or international? And if it's US-based, any clinic sites that you've chosen?
Yeah, not yet. Preliminary to say, obviously, we're planning ahead, and it may be that it will be both US and international. We'll see how this evolves, but we have not disclosed details. And of course, as the public community is very well aware, all these studies need to be vetted by the regulators and ultimately by the institutional review boards, et cetera. So of course, what we're talking about here today is the general plan, but this will all be scrutinized very closely by multiple layers, including us, of course. We're very careful about how we design these things.
And do you know yet whether, if somebody participates in a phase 1, do they have the opportunity of being a member of multiple cohorts? So going from single ascending dose to multiple ascending dose, or phase 1 to phase 2, or will there be a chance for multiple participation? Yeah,
we can't exactly precise what that is, but the intention to date is that people who participate, let's say, in a single ascending dose could potentially participate in the multiple ascending dose and potentially in phase 2. Yeah, given that this is a small molecule that once given, there's an effect that lasts for a while, but it's not forever, right? Eventually it will wane. So, ultimately you'll have to repeat dosing. We just, our initial plan is to dose once weekly. These are injections, right? Not prolonged infusions. We obviously will have to see how long the effect lasts for before we make a plan of how often we dose. It's possible that you could dose less often, but of course, this is something we need to test.
Along dosing, along that vein of questions, there are a few questions related to that. And one question is, why is this an IV dose and not subcutaneous or like a pill? And I think you spoke to, again, how long does the dose last, but how long does the infusion take? And can you kind of give folks an idea of what that might look like to have an infusion? Do you have to go to a hospital for that? Is it something that's done in an outpatient clinic or at home?
I can take that one. It's an injection. It's not an infusion, and it takes about, we hope, less than 5 minutes altogether. The challenge with the molecules is to find a mechanism to formulate it, which means to prepare it so that the body can receive it in a manner that's easy to administer. And so when you first start out with molecules, you often are forced into, in order to accelerate the program and get it to the clinics, it's often necessary to formulate it in such a way that you administer it intravenously. And that's what we're doing. Whether or not the molecule can be formulated for other approaches remains to be determined, and we're working hard on figuring out how it might be done otherwise. But this early in the program, it's not uncommon to have an IV formulation.
Yeah, and as far as the site of administration, of course, we'll have to see how well tolerated everything is. But assuming the tolerability is what we've seen in these animal studies, then at some point, perhaps after the initial injection 1 or 2, then this could be done as an outpatient. And I think even if we were to remain with an intravenous formulation, there's a fair amount of precedent of commercially available successful drugs for a variety of diseases that are in fact delivered intravenously, especially now with a much more widespread ability to, say, visiting nurse to come to somebody's house and administer something intravenously. So that's evolved quite a while, such that is very, not uncommon to have, even in neurological diseases, to have intravenous injections.
And in some of the animal studies that you're doing, like the one that you shared with us, where you were looking at where the gene tacks go in terms of the brain and spinal cord, are those animals also dosed IV?
Exactly, yeah. They're supposed to. So what we test in the these animal studies is what ultimately will be intended to be used in humans. Obviously, we test in animals much higher doses than we need to deliver to humans, just to push the envelope. But the route of administration and the type of formulation are similar.
Yep.
To what would be given in humans.
And can you give our community an understanding of, in those safety studies in the animals, like how is safety assessed? Is it assessed, in the clinic and then histologically? Like what range of studies are done to look at and define safety?
Do you want to answer that?
Sure. Yes, it's a very detailed protocol. All the animals are studied exactly like in humans. Their behavior is observed. Their vital signs are measured. Bloods are taken to look at liver, kidney, and other functions. After they finish the protocol, a group of them are set aside to make sure that if they do find something after X number of weeks of treatment, that's called a recovery group. So you stop treating them for some time and look at the difference between those that have been treated for, let's say 13 weeks, and then you look under the microscope and do histopathological evaluations of all the tissues. And then the total package is reported out in a very large series of data points to make sure that there is no findings that would preclude, that would be concerning in terms of safety. We look very much at the liver, the lungs, the heart, and the kidney, just to mention a few, but it includes every organ in the body. And it's a very standardized and very rigorous approach. With the number of animals, it's quite a lot, to be honest with you.
So along with the histological evaluations that you're doing in the animal studies, have you been able to look at any protein levels in any of the organ tissues?
Yeah, let me answer. Yeah, so the healthy animals don't have deficiency in frataxin, right? So that obviously is not helpful. There's several I've seen on the Q&A here. There's several questions regarding animal models, and it's all this. As a clinician, former clinician, that is, I'm no longer a clinician. I'm always surprised how much there's interest in animal models when ultimately we're testing in human cells. But nonetheless, it's a legitimate question. So very few animal models, in fact, I think I know at least one, maybe two, I don't know, others might know more, have in fact recapitulated the human disease in terms of the actual mutation, right? There's the gene tag only works when there's a GA repeat expansion that is blocking transcription elongation. So all the other models wouldn't be useful for gene tag because they don't have the GA repeat expansion. There's one model that I know FARA's worked very closely with Mark Pook, the Pook 800J mice, again in mice, these are not in non-human primates, where they were able to insert a mutant human gene in a background of a mouse genome. So there starts to be sort of a bit of a chimera there, no pun intended here, where you have the expansion that is from a human normal mutant gene inserted in randomly in the mouse genome. So that's not ideal, and these animals also have relatively few symptoms related to FA, so they're not something that we could measure, clinical benefit very reliably. Nonetheless, in the early days of design, and FARA, well aware of this, there was a lot of interest in testing our prototype molecules in these animals, and lo and behold, with systemic delivery of gene tags, not this particular gene tag, but some other gene tags which are even less potent in these molecules, we were able to measure increase not only of mRNA, but also protein in both brain and heart of the Pook 800J mice. Very clear increase in protein levels in an mRNA. So the long explanation to say, yes, it does increase mRNA and protein in a specific mouse model that has GA repeat expansion, although this is a far from ideal model for drug development, right? In drug development, we need something that is very discriminating, that you can test a dose-response relationship, duration of effect in some way that is more consistent. And for that, it became quite clear that using cells from patients that were donated from patients was much more appropriate, and that is exactly the cell model and the genetic makeup that ultimately we're intending to treat in actual people who have FA. So we feel that this is, from the point of view of the fine-tuning and selecting very specific molecules, this was much more discriminating and useful as a model than were the Pook mice. And this is not to say the Pook mice are not a good model. It's just saying that we're not found to be very useful in the process of triaging and selecting specific molecules.
Yeah. Thank you for that clarification. I think you're right. A lot of people were concerned and are concerned about the animal models, and it's a different beast in this particular approach. Jen, any other questions that you're seeing that we should?
Let's see. We answered some of these. There are a lot of questions kind of going back to the clinical trial. There are some questions and some requests. I'm sure you're seeing there's interest for maybe expansion of the age criteria. There's also a question just to clarify, if someone is using a wheelchair but can transfer independently, would they potentially qualify for inclusion?
I can answer that. Right now, we're trying to keep the inclusion criteria as open as possible. So I think if somebody could be in a wheelchair and transfer safely and be able to undergo the duration of evaluation that we're planning, that would make perfectly reasonable sense. So,
yeah, just a disclaimer here, and I always have to. And Matt's a clinician, I'm a clinician at heart still, and oftentimes we want to answer these questions, but the reality is that the investigator ultimately is the one who's responsible for doing once you have the protocol written, ultimately is at the discretion of the clinical investigator to select who are the patients that are most suitable entering the study. And I think, I can imagine that there's obviously a lot of interest in participating in a trial like this. We hope that if we can show it to be safe and well tolerated, that we'll continue to, enroll patients, and perhaps, further along the development, you can actually see potentially expansion a little bit of the inclusion criteria, right? So the first, I think that we feel very responsible, one, for getting this compound into the clinic in the safest possible manner that would enable us to actually test whether or not this molecule is well tolerated, and two, whether it can restore frataxin. Then if we have those data in hand, there will be a much greater impetus to then, progress with the program. But this initial step is critical to make sure that if we get into the clinic in a way that is as suitable as possible. I see some lightning outside of your window, Jen.
Yeah, my house just shook. Like, oh, sorry.
Wow, that's a big storm.
Yeah. So if I disappear, you know why.
Hopefully not.
But, along those lines, I think some of the questions too coming in were, about 5% of people with FA have the GAA repeat expansion on one allele and a point mutation on the other allele. And so I think there's interest from that community to have some understanding eventually of, whether or not gene tags could also work when you only have one expanded allele and not two.
Yeah, we'll seek to also help answer this question probably in the first, in very initial trial, but it's something that has arisen as a question, which is a legitimate question, one that we'll look into.
We'll have to do specific assays on cell types like that to see if how the drug might work.
There's a question to ask for some more information about the mechanism of action as it relates to similarity to like an HDAC inhibitor. Is it working independently of heterochromatin? Is it a methylation, acetylation? If you can just review quickly the mechanism of action again.
So I think that perhaps the best source there would be to read the paper in Science by Irwin et al., 2017. I think that you get all the answers that you're looking for there in a much more elegant and eloquent way. But they are different, and I think most importantly, the order of magnitude of response to a gene tag versus an HDAC inhibitor are very different. This is an order of magnitude difference in terms of the effect. And we'll present data showing these results in the upcoming, hopefully in the upcoming conference. So,
and then just going back to the clinical trial design. Have you determined all the clinical outcome measures that you will be using? There's an assumption that mFARS would be one of those measures, but are you planning to look at any others?
Well, as a phase 1 trial, we're really not going to look at clinical outcomes.
Right.
The point of that is safety, number 1, pharmacokinetics, number 2, and yes, pharmacodynamics in terms of the frataxin levels in the peripheral blood monocytes.
We'll measure some of the outcome measures at baseline to characterize the patients and in what conditions they start out the trial, but it's unrealistic to expect that after a single dose you would see a change in mFARS or other things, right? So this will be reserved for a bit longer treatments.
And I just wanted to address, there are several questions asking about how people can participate, when would they be able to enroll? So this is an ongoing design for the study, and communication will continue through FARA to let the community know when screening is open, eligibility is being determined, and so we will continue to communicate the availability of the trial when it's ready to recruit and enroll.
Perfect.
I know this, I'll ask this question. I know it's really hard to say, but there's concern, questions, curiosity. If Reata's drug, amoxicillin, is approved before or during this trial, how does that affect eligibility or ability to participate in your clinical trial?
Yeah, very early on in the phase 1, especially in the initial stages of phase 1, we tend to try and keep confounders away. So that could be considered a confounder. But in the long run, hopefully the Reata drug will be approved and will be useful and helpful to patients. Then, of course, clinical trials that come, after the approval will have to incorporate an ability for patients on the OMAV to participate in trials. But it has to be done safely, perhaps a better understanding how they might or might not interact and whatnot. So these are things that we'll have to look into.
Mm-hmm. Thank you. Yeah, there's a lot of uncertainty around that question.
And that's not just to reassure everyone, this is not unusual, right? In most clinical trials, we have to contemplate, ability of patients who might be taking different drugs of all kinds, in fact, including supplements and other drugs and analgesics and others into the clinical trial that you're testing for a novel compound. So you'd have to do it properly, but this is a reality we'll have to address.
And there are ways to address that. And I'm sure we'll have ways to make sure that's done in a fair and equitable manner, as well as in a scientifically rigorous manner.
Mm-hmm. Along with that, there's a question about currently participating in clinical trial and being able to also participate in this one. And so I can answer that in terms of when there's an investigational study going on, a participant can only be involved with one trial at a time, because as Dr. Seifert mentioned, we don't want confounding effects when you're looking at either the safety or the efficacy of that investigational compound.
Well, this has been great. As you can see, there's so much interest from the community, lots of questions, lots of enthusiasm and discussion. And, I think a few folks are now putting in their comments of thanks for your, hosting this webinar and sharing all this information with us. And Kyle, I'll end with maybe his question, which is there anything that we can further be doing to help you and help the program move forward? And we FARA, the FA community.
You have, in fact, we've talked about this with the FARA colleagues, that part of why our co-founder initially looked at Friedreich ataxia is because he had received a FARA grant, actually a Kyle Bryant grant, I think, research grant. And so that steered his research into Friedreich ataxia and hence design. And ultimately, design was founded. We had received a grant from FARA too as design. And I think these are ways to encourage and support early research. And of course, at this stage now, we perhaps less in need of FARA's grant support, but certainly we have received tremendous amount of support from the community and a partnership. Of course, any time you're developing a new pharmaceutical, it has to be a very collaborative effort between the company that is developing this, obviously patient advocacy groups and the patient community, because there's ultimately we talk about testing in humans. So that's a very sort of a careful, involved path, and then this partnership is critical for the success and to help test this. I think it's critical that we test this, that we see if it works as we've seen working in these cells and in animals. And that's, I think, the greatest contribution. So I appreciate the support.
Thank you. We look forward to continuing this collaboration, and very, very thankful for doing this informational webinar It gets the community to understand and get excited about the potential trial. It also allows us to use this recording to bring other people who then become interested in the trial to be able to review this material. So it goes a really long way to continuous engagement with the FA community. So thank you.
Yeah, and we'll keep everyone, of course, keep FARA and keep everyone apprised as we make progress.
Great.
Indeed. Thank you.
Good. Thank you for joining us tonight. We're going to wrap up. This recording will be put out through the FARA website in the next couple of days. And certainly, if you have any additional questions, you can always email the FARA staff at info@curefa.org, and we'll be happy to get back to you and let you know what answers we have. Thank you and good night.
Good night.